Ultimately, robots are just a bunch of metal and motors. Software must orchestrate the processes involved in conducting an experiment. Some of this software will be encoded in the instrument's firmware, and other times you control the instrument with an attached computer running software within normal operating systems, usually Windows-based. However, the automation of experiments is not limited to operating robots, so let's take a look at the other types of software used for automating synthetic biology research today. Most software associated with synthetic biology is associated with standards development, automated design, or computer-aided manufacturing. If I tell you to go build a part out of GFP, you pretty much understand what I mean, but you must inject a great deal of knowledge and make many micro decisions in order to implement the experiment. For example, should you make a BioBrick part or a Golden Gate part? Or can you do anything you want? Which GFP? What codon usage should I make the GFP out of? In what form do you want it? Just as an APE file, or do you mean a PCR product or a clonal DNA sample? There is a lot of subtlety to these terms that usually is ignored when we speak about things like parts and genes. When you write software, the computer only understands what you teach it, which requires that you very carefully define these concepts in code. This is the essence of standardization as it applies to BioCAD tools. The BioBricks idea is essentially to define a series of rules for the contents of a sequence to simplify what a computer must be told in order to understand the usage of the DNA fragment. In BioBricks, these rules are statements about forbidden restriction sites and how to calculate a larger composition sequence from a list of constituent basic parts. The SPOL effort is a consortium of researchers that is developing a series of these standardized concepts, primarily focused on the representation of annotated DNA sequences and graphical representations of these sequences. Automated design tools allow a user to consider all possible options for implementing a genetic device. If I told you to make a pathway for xanthophyll, you'd probably Google xanthophyll biosynthesis and find some pages talking about how it is made naturally. You might poke around and find some additional ways to make that chemical, but you never know for sure that you considered all options. If instead you provided all the raw data in a standardized form to the computer, you can use algorithms to search the space for all possible solutions. Such approaches have been used for anabolic and catabolic pathways, but they are also under development for genetic circuits. Automated manufacturing, or CAM-based tools, employ standard assembly protocols like BioBricks to design a fabrication process. So you input a specification of what DNAs you'd like to put together, and these software tools figure out how to piece that together most efficiently and cost effectively. Some of these tools connect directly to liquid handling robotics to fully black box the details of DNA fabrication. There are a variety of pre-existing tools adapted from fields